Hi, everyone, and welcome to Chapter 14. A few announcements. Again, as I always do, if you are watching this in the fall of 2020, then you have an exam coming up this Friday, okay? Let's see, what is the date on this Friday? I believe it's October 16th. This exam is going to cover Chapter 11 all the way through Chapter 14, okay? So today we are going to be switching gears and we're going to be talking about Mendel and the gene. So in other words, we're going to be talking about genetics. This chapter I find is one that many students enjoy because they have some level of familiarity with it. If I were to throw terms at you like homozygous, heterozygous, Punnett square, then most likely you would know what I'm talking about. So I'm going to be dividing this chapter into two sections, as I often do. The first section is going to be a lot of review of general genetics. And then the second section will talk some more about some exceptions to what we think of as general genetic rules. So for the first part of our PowerPoint, we're going to talk about Gregor Mendel and his experiments. And we're going to look at Mendelian genetics, looking at just one trait at a time. And that is actually something called a monohybrid cross. And we'll go over this again. And then we will switch to talking about two traits at once. This is something called a dihybrid cross. So we're going to review the experiments that Mendel performed to figure out some of the rules of inheritance. And then we're actually going to name what those rules are that Mendel figured out. There are 4 of them. The next thing we're going to do in the first part of this PowerPoint is we're going to tie these rules back to what we learned in Chapter 13 about meiosis. And then finally, in the second part of the PowerPoint, we're going to go over the discovery of chromosomes as genetic material and extensions to Mendel's general principles. All right, so let's talk a little bit about heredity. So I kind of want to get you in the mindset of the time in which Gregor Mendel would have been working. So many of you probably have heard of Gregor Mendel. He's often considered the father of genetics. He was working in the mid-1800s. And at this time, there was a lot of interest in how traits were inherited from generation to generation. This makes a lot of sense if you think about it, because at the time, there was a great deal of farming going on, right? So we're just beginning the Industrial Revolution in the mid-1800s, and primarily we're an agrarian society at that point. That means that if you're a farmer, you're interested in the best possible crops you can grow or the best livestock that you can manage, okay? So there was a lot of interest in how to grow the crops the best that you could. And in thinking about how farmers might grow good crops, there were a couple of hypotheses about how traits were passed from parents to offspring. So like if you have a particularly hardy strain of tomatoes and you're a tomato farmer, you might want to create offspring from your tomato plant that are also particularly hardy. So it would be important for you to understand how things are passed on from parents to offspring. And as I mentioned, there were 2 really prevailing hypotheses about inheritance at the time. The first one is something called the blending inheritance hypothesis, okay? Now, this one probably makes good intuitive sense to you even today. So let's think about the blending inheritance hypothesis. And to explain this, I often like to think of a dog called a Labradoodle. Many of you have probably seen a Labradoodle, and if you haven't, you can Google it. But a Labradoodle is a dog that is a cross between a Labrador retriever and a poodle. If you look up Labradors, and you look up poodles, and then you look up a Labradoodle, you can see that a Labradoodle is a pretty good blend of those 2 breeds, okay? This also is kind of true of people, right? So if you have a friend that, you can say, oh, well, I can see that you have your mom's eyes, or maybe you have your dad's big ears or something like that, right? So this kind of made intuitive sense to people that traits blended between parents into their offspring. The second big hypothesis of the day was something called inheritance of acquired characteristics. And this idea was really championed and well articulated by a guy named Jean-Baptiste de Lamarck. And I'm just going to make you know his last name, Lamarck. And actually, at about this time, this is about the time when Darwin was working and really promoting his theory of evolution. And so the idea of evolution really battled this idea of Lamarckism, which is inheritance of acquired characteristics. Now, what does that mean? Well, let's take a look at this sort of giraffe thing. But before I do that, let me go back up here. This is a visual on what the blending hypothesis might look like, okay? So I used labradoodles, I used people as examples, but here you have a red flower and a white flower. And if the blending hypothesis is true, then what you would end up with is a series of pink flowers if these are the two parents. So that's the blending hypothesis. Now let's take a look at the inheritance of acquired characteristics hypothesis. So in this famous example, you have giraffes, right? And the idea is that the original giraffe was not all that long in the neck, but that this giraffe would most likely seek leaves toward the top of the trees where they're quite tender, okay? And in seeking those leaves, this giraffe would extend its neck, and maybe throughout the course of its lifetime, its neck would be stretched a little longer than it was ordinarily. The theory of acquired characteristics suggests that once that giraffe here was able to stretch their neck just a wee bit, that they would then pass that newly stretched characteristic onto their offspring, who in turn would work throughout the course of their lifetime to stretch their necks, to eat the leaves in the trees. That individual's neck would get to be a little bit longer, and they would pass that on, et cetera, until you have the giraffes we see today. So to summarize, the theory of acquired characteristics suggests that the reason giraffes have long necks is because previous generations worked so hard to stretch their necks that they acquired this characteristic over time. Now, this hypothesis probably doesn't make a lot of sense to you, but it was at the time, remember, this is pre-evolutionary theory being part of the conversation. So folks were trying to find ways to explain some of the more outlandish traits that they saw in animals. But another way to think of the inheritance of acquired characteristics would be to think of a human being, perhaps someone that spends a great deal of time in the gym, and they become very muscular. They acquire this very muscular physique, and they then pass that on to their offspring. So I always call this sort of the swole baby idea. You probably haven't ever seen a baby that was super swole and had lots and lots of muscles. So today we know that the inheritance of acquired characteristics hypothesis is not really true, although if you take genetics with me, we will talk about a phenomenon called epigenetics, where we're beginning to find out perhaps it's more correct than we think. Epigenetics. Okay, but for purposes of this class, just know we don't really believe that anymore. At the time that the time that Mendel was working, these two things were definitely hypotheses, and I would argue that this is probably the one that most farmers subscribed to. So Gregor Mendel was a monk, and he actually, I have here 1865. This is the year that he published his work. He probably began working on his experiments somewhere around 1856 to 1859, okay? So he lived in the Czech Republic. He was educated there. He went to university. He had actually aspired to be a teacher. But as part of the monastery, monks had various jobs that they were assigned, and Gregor worked in the monastery gardens. And so he worked with a lot of different plants, but he worked pretty closely with pea plants, okay, to grow peas as crops for the monastery. And he started noticing that peas have several traits I have them written down here as polymorphic, but in particular, these traits are what I would call dimorphic. Now let's break this word down. A dimorphic trait. Morph means shape and di means two. So a dimorphic trait is what I call an either or trait because it only has two states. Here's what I mean by that. If you're growing peas, they can be pink or they can be white, okay? The flowers of the pea plant can be pink or white. There aren't any sort of red or orange or anything like that. If we go down to a slide that I'm going to show you in a minute, you can see that the color of a pea can actually be yellow or it can be green. The shape of a pea can be wrinkled or it can be round. The pod can be inflated or constricted. Do you see that we have a whole bunch of traits that are an either or kind of scenario? So Gregor Mendel noticed this and he thought that a pea plant would make a great model organism. So what is a model organism? A model organism is one that is used in research as a test case to discover general principles. So the pea plant is being used as a test case in this scenario to investigate inheritance patterns because Gregor Mendel knew that he could follow these either or traits pretty easily, okay? Another model organism that you might have heard of is the fruit fly, which we're actually going to talk about in this lecture. In my field, a model organism would be the zebrafish. We use that a lot in research. Mice are a model organism. So model organisms are typically organisms that are easy to cultivate, so they're easily grown or easily managed in a laboratory. And they're ones that have short generation times that you can work on over and over again to investigate various scientific phenomena. So our boy Gregor, he's doing his work on pea plants and he's following them because there's several either or characteristics that he can follow as he moves through his work, okay? There's our characteristics. Note that they are all either or. And another way of saying that is that they are dimorphic. And we're going to come back to this slide in a second. So here is what Gregor decided to do. He started out by establishing what he called pure breeding plants, okay? So what do we mean by that? Well, in this scenario, let's look at the shape of seeds. So here we have seeds that were produced by two lines of plants in Gregor Mendel's garden. One line of plants produced round seeds, and the other line produced wrinkled seeds. Now, what I mean by pure breeding is that he developed a strain of pea plant that always produced round seeds. So in the first generation, he had round seeds. He allowed that plant to self-pollinate. It made more round-seeded plants. He allowed those plants to self-pollinate. And over several subsequent generations, he was able to establish a strain of plants that always, always, always produced round seeds. Put a pin in that. That's going to be important later. He did the same thing for wrinkled seeds. So these were plants that, initially he found wrinkled seeds. He allowed that plant to self-pollinate. It made more wrinkle-seeded plants. And over several generations, he made plants that always, always, always made wrinkled seeds, okay? So these were what he called pure breeding plants. Now, remember that I told you that the theory of the day was primarily, I would say, the idea of blended inheritance. So what Gregor decided to do is to take his round-seeded plants and have them mate with wrinkle-seeded plants. And what he predicted based on the idea of blended inheritance was that those offspring would all have sort of an intermediate physical appearance, okay? They would have slightly wrinkled seeds. And if he allowed these plants to self-pollinate, then they would produce more slightly wrinkled seeds. So he decided to carry out this experiment. Here are our pure breeding plants. He allowed them to mate, and lo and behold, what he found was not wrinkled seeds. Instead, he found that all of the baby plants produced peas that were completely round. In other words, what happened was this physical appearance completely disappeared in this generation. Now let's get some terminology out of the way. This generation is the parent generation, and it's often abbreviated P. Now you'll notice that this generation here is abbreviated F1. What that stands for is the first filial generation. Filial. And that is from Latin, okay? Filial means like the first son generation or the first offspring. So F1 is your first generation from the parents. F2 is your second generation from the parents. So it'd be the second filial, okay? So here we are in our F1 generation, and I want you to notice something. One of these physical traits has completely masked the other trait, hasn't it? So this trait has completely obliterated this trait, and all we have are round seeds. So this was a very curious result, and what Gregor Mendel then decided to do is he said, okay, this is weird. I'm going to now allow these plants to mate amongst themselves. I'm going to let these F1 plants cross. So we're going to have an F1 that is round cross with another F1 that is round. When he did that, what he found was that the wrinkled appearance returned, okay? Now let's get some vocabulary out of the way, and I'm going to actually pull up my notes because I have a whole series of vocabulary I want to talk about. Oh, maybe it's not on this slide. I keep saying appearance, but I'm going to start using the word phenotype. Phenotype. PH, phenotype, physical characteristics. So the phenotype of an organism is the physical characteristics associated with a trait. So we have a round phenotype for seed shape, and we have a wrinkled phenotype for seed shape. Seed shape is the trait, but the phenotypes are round and wrinkled. So in F1, we got rid of the wrinkled phenotype. In F2, the wrinkled phenotype reemerged, and it reemerged in a very specific ratio. There were 3 times as many round seeds as there were wrinkled seeds. That was quite curious. So looking here, he wanted to know, let me go back. So he thought this was weird, okay? This was very, very strange. So what Gregor Mendel decided to do was to look at other traits. He looked at seed shape, and he found that when he got to the F2 generation, there were 3 times as many round as there were wrinkled seeds. Then he looked at seed color, and he found that when he did the same experiment and got to the F2 generation, there were 3 times as many yellow seeds as green. He looked at pod shape, 3 times as many of this one, pod color, 3 times as many of this one, flowers, growth patterns, height. For all of these here, he found that there was a trait that was 3 times as prevalent as another trait. All right, let's go back to the previous slide. So I mentioned that we had one trait, let me leave that there, that masked another. You guys probably already know that a trait that masks another is called dominant. And the trait that is being masked is called recessive. Okay, so what happened here is a dominant plant mated with the recessive plant and produced all dominant offspring. Now, to explain this, what Gregor Mendel decided was that each of these original parent plants must have 2 copies of what he called a factor. because at this time, we did not understand about genes. So he said, each of these parent plants must have 2 copies of a factor for seed shape. And because these are pure breeding plants, I'm going to say that these 2 copies must be identical. Okay? Because these are pure breeding plants, we've got 2 copies that are identical for round seed shape and 2 copies of a factor that are identical for wrinkled seed shape. Then he reasoned that what must happen is that in order to produce these offspring, these copies must separate, and this parent gives the offspring 1 copy, and this parent gives the offspring 1 copy, and you have these babies here that have 1 copy of each factor for 2 different traits, 1 dominant copy and 1 recessive copy. And he called these resulting offsprings that had 1 of each, he called them hybrids. So he called these pure breeding and he called these hybrids. I'm going to introduce a new set of terms for you guys, and I'm going to clean this up a little bit so you can see what we're doing. I'm going to say that these folks that have 2 of the same are going to be called homozygous. And these that have 2 different are going to be called heterozygous. And that's going to be easy for you to remember because the prefix homo means same and the prefix hetero means different, okay? So if you're doing a genetics problem, say on an exam, if I say the word pure breeding in the problem, that is a synonym for homozygous. So you can be homozygous dominant, like this guy here, or you can be homozygous recessive, like this guy here. If I say hybrid, that is synonymous with heterozygous, meaning you have 1 copy of a dominant and 1 copy of a recessive. Okay, so now what's happening in this generation? So we've crossed 1 of these with 1 of these, so 2 of these little critters here. And in order to understand what happens here, the best way to describe that is using something called a Punnett square, okay? Punnett squares were devised actually in 1902 by a guy named R.C. Punnett. And what you're really doing when you draw a Punnett square is you're figuring out probabilities of offspring being produced. So let's walk through this. I told you that we have these parents that are homozygous. Oops. And that these guys are heterozygous. Now, one of the things that Gregor Mendel predicted is that at some point, when these critters go to mate and create an offspring, these factors must separate. Now, if you're paying attention, hopefully this reminds you of meiosis. Remember that in meiosis, you take 2 copies from a diploid organism and you separate them to make a gamete that only has 1 copy, okay? Here you have 2 copies from a diploid organism and you're separating it to make a gamete that only has 1 copy. So if we undergo meiosis here, which again, Gregor Mendel did not know about, but if we undergo meiosis here, we separate out this copy and this copy. This seed here is capable of making gametes with a big R or gametes with a little r. So let's put those on our Punnett square. What you're really putting on a Punnett square when you do this is you're putting down what went into the gametes, okay? So these are the gametes from 1P, and we're going to cross that with gametes from another P. Actually, let me back up. I have 2 Punnett squares here because I wanted to show you twice what was going on. Let's start out with the parents, okay? you have two gametes that you can form here. You can have a big R gamete or a big R gamete. Those are the only two options because this individual has two R's. You have two different gametes you can form here, little r or little r. So the resulting offspring are going to look something like this. One parent can donate a big R, one can donate a little r. There you go. Big R, little r, there you go. Big R, little r, there you go, and there you go. So there are four different possibilities that you can have, but they're all the same. So the probability of coming up with a big R, little r offspring is 100%, right? Actually, when we deal in probabilities, we usually don't deal in percentages. We actually say that probabilities go from 0 to 1. So this is a probability of 1. So this is what would happen in the F1 generation. So these are our parents here. This is our P generation, and these are the result that would be in our F1 generation. So parents, F1 generation. Now let's take two of our F1 seeds and let's to figure out what their gametes might be. So you can have a big R, or you can have a little r, big R, little r. All right, what are our possibilities here? If these two combine to form an offspring, that P is going to have an RR makeup. If these two combine, it's going to be big R, little r. These two, big R, little r. And this one's going to be little r, little r. Now let's take a look at what's going on in each of these. If you were to tell me what the phenotype, what the physical appearance of this individual would be, this guy is dominant to this guy, and we have two of these. So we're going to have the dominant appearance, which is round. So this is going to be a round seed. This is dominant to this guy. This is going to be a round seed. This is dominant. This is going to be round. And these are actually two recessive copies. So this is going to be wrinkled. Now let's look at what's going on probability-wise. You have out of 4 options, 1, 2, 3 of them are round and 1 of them is wrinkled. This explains the phenomenon that Gregor Mendel observed of a 3-to-1 ratio. Now, I mentioned the term phenotype before, which is physical appearance. Now let's contrast that with the term genotype. The phenotype is the physical appearance. The genotype is the genetic makeup. So, what we have here is a 3-to-1 phenotypic ratio. In other words, we have 3 dominant physical appearances and 1 recessive physical appearance. Now let's see if we can figure out a genotypic ratio. Okay, so here you have a dominant genotype because you have this genetic makeup and this genetic makeup. Here you have a dominant recessive genotype, dominant recessive genotype, recessive genotype. So your genotypic ratio is 1 to 2 to 1, okay? That's your genotypic ratio. Because there's 3 different genotypes, 3 different genetic makeups. Now there's one more set of vocabulary that we really need to know in order to understand genetics, and that is the idea of genes versus alleles. And I'm definitely running out of space here. Okay, So a gene codes for a trait. In our example here, the gene codes for seed shape. An allele is a different version of that trait. So our alleles here are round or wrinkled. Both of those code for the trait seed shape, but each of these is a different version of seed shape, round or wrinkled. Now remember, at the time that Gregor Mendel was working, we hadn't worked out genes or alleles, but it's easier if you understand those concepts moving forward. So I'm going to insert a new slide here, and I'm going to make this slide blank because I want to just go through the vocabulary just to make sure that everyone understands the vocabulary. So things that you need to know are going to be the terms homo and heterozygous. Okay, and I'm going to put zygous here because they're both the same. Make sure that you understand that. And make sure that you understand that there are some other things that mean homozygous, like pure breeding. And heterozygous would be hybrid. Okay, so make sure that you make those connections. I want to make sure that you understand the difference between a phenotype and a genotype. Make sure that you understand the difference between genes and alleles. And make sure that you understand the difference between genotypic ratios there we go, and phenotypic ratios. Okay, so satisfy yourself that you understand all of these things. Now I'm going to back up for a second because there's a slide I skipped. So a few things, we've gone through Gregor Mendel's work. A few outcomes that I want to point out. First of all, his blending hypothesis is not supported. His data do not support that hypothesis. Let's go back and look at one of my very messy slides here. If you look, let me go here, oops, and I do a little erasing. If you look here, boop, 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 boop. This guy here is not a blend of these two guys, okay? So the blending hypothesis is not supported, okay? That's one thing that you can get out of his work. The second thing you can get out of his work is that he concluded that inheritance must be passed on by particles that remain intact. So in other words, if we go back up here, oops, let's see this one. He said, oh, they must be passing on a factor. The R is our factor. He didn't know that those factors were genes, but he knew that there was something getting passed on. And he said that, that each individual must have 2 copies of those factors and that they separate during the formation of eggs and sperm, okay? So let's go back here. Oops, sorry, go back here. So 2 copies of the factor and they must separate during the formation of eggs and sperm so that the gametes can fuse to form a new baby, okay? So this is all stuff that we've already learned. So here, it all comes back to meiosis. What Gregor Mendel really did was he kind of predicted the outcome of meiosis, okay? So just as a quick review, this is before the cell divides, and we know that this would actually be chromatin, but let's just, for the sake of argument, This is a pre-division cell. It is a diploid organism, right? So this organism is diploid, 2N. So we have one homologous chromosome here and the second homologous chromosome here. The blue came from dad, the red came from mom. Maybe this one has a big R and maybe this one has a little r, okay? Now, what happens during the S phase of the cell cycle is that these chromosomes, the DNA on them becomes duplicated, right? So here we are, the DNA has become duplicated and we have two copies, R, 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 and I'm gonna put this R down here. Okay, now we undergo our first meiotic division, meiosis 1. So here we go, little r, big R, big R, little r, big R, big R. So in this scenario, we started out with a parent that was this, and we ended up with some gametes that were either this or this. The same thing that we're seeing in our Mendelian genetics. So basically, he called this phenomenon the principle of segregation. Because the term segregate means to separate, right? Okay, the principle of segregation. So let's kind of summarize some of the classic conclusions that Gregor Mendel arrived at as a result of his experiments. So the first one is he concluded that there must be two copies, which he called factors, but we're going to call genes, for each trait. So what he really figured out was he really figured out the concept of diploidy, right? You have two factors of each trait, one from your mom and one from your dad, okay? The second thing that he figured out was that these factors separate to make gametes, okay? And they do that during meiosis. So he called this the principle of segregation. Oops. And then the final thing that he figured out is that one of these factors masks a second factor, doesn't it? So he also figured out something called the principle of dominance. Where dominance, oops, hold on, let me spell that correctly for you. Whereby one factor or one gene masks the other. Okay, so that's where we are right now. So Gregor Mendel had done all these experiments using one trait, but then he decided that what he would like to do is to look at two traits together simultaneously, okay? And what he was specifically trying to figure out in looking at two traits together is whether you inherit traits as a unit or whether you inherit them independently. And I'll explain what I mean by that in just a second. But before we move on, let's get some more vocabulary out of the way. If you're looking at one trait at a time, we call that a monohybrid cross, okay? If you are looking at two traits simultaneously, that is what we refer to as a dihybrid cross. Okay, so what Gregor Mendel wanted to know is whether you inherited traits in a chunk, so to speak, or whether you could inherit them independently. So to illustrate this idea of what he was trying to test, Let's go back to our seed shape example, and this time let's add in a second trait of color. Now, just to kind of get us on the right track, remember that for seed shape, round, which we're calling big R, that was dominant to wrinkled, which we're calling little r. For seed shape, we're going to say that yellow is dominant, or sorry, for seed color, is dominant to green, okay? Now, hopefully you're noticing something, and this big Y is yellow, and the little y is green. Now, hopefully you're noticing that whenever I designate a dominant allele, I use a capital letter. When I designate a recessive allele, I use a lowercase letter. And that is pretty much standard, especially if you're talking about plant alleles. When you get to human genetics, sometimes the designations we use are a little different, but in general, if you see a capital letter, know that you're dealing with a dominant feature, and a lowercase letter is a recessive feature. Now, what Gregor Mendel wanted to know is, again, whether you inherit things together. So, for example, here we have a P that is wrinkled and green, and a P that is yellow and round, okay? So let's put that here. We've got a wrinkled and green, which would be little r, and then green would be little y, right? And so I'm just going to write out the phenotypes here. So that is wrinkled and green. And we have a second P that is round, round and yellow. So what Gregor Mendel wanted to know is, do the features wrinkle and green always get inherited together? Do the features round and yellow always get inherited together? Or could you mix and match this? Could you have a wrinkled P that was yellow? Because that would be different looking than the parents, wouldn't it? Could you have a round P that was green? So in other words, if traits travel together, then you're only ever going to have the phenotypes that you saw in the parents, wrinkled green, round yellow. If the traits mix and match, that means that they travel independent. Wrinkled can be with green, or wrinkled could be with yellow. Round could be with green, or round could be with yellow. So that is the next set of experiments that our boy Gregor sought to do. If you were to have a situation, which they're calling dependent assortment in your book, and I'm just going to say traits traveling together, here's kind of what you would expect. Okay. Here we have our wrinkled green, round yellow. They make round yellow offspring. Okay. This is the F1 generation. And if you can only have parental phenotypes, then in the F2 generation, what you would expect if you crossed two of these guys together is a 3-to-1 ratio of the round yellow dominant phenotypes to one recessive wrinkled green phenotype. Now, so that's kind of what I'm talking about here, okay? This is not what our boy Gregor found, okay? So let's go back here and look at this experiment again. So what our boy Gregor did is he had wrinkled green, round yellow, and this part was true. Yes, in the F1 generation, we ended up with all of them being round and yellow. Okay, but what was happening is when he would do this cross here, where he would take two of these guys and cross them against one another, he found a phenotypic ratio of 9:3:3:1. And I'll explain this in just a second. Let's go back here. Let's think about this parent here. This parent can donate this little r or this little r, this little y or this little y. So this parent can only donate a little r or a little y to the offspring. This parent can donate this big R or this big R, this big Y or this big Y. So all that this parent can donate to the offspring according to the rules of meiosis is a big R, big Y. If you put these two together, what you get is big R, big Y, little y. Now, here's the key. What is happening when we cross two of these? And I want you to hold this ratio in your mind because I'm going to demonstrate how we get there. So we're in our F2 generation now, and we have two of these characters here, big R, big Y, little y, okay? And it turns out that what happens is something called independent assortment, where yes, you can donate a big R or a little r and a big Y and a little y. But in thinking about how these R's and these Y's combine in the gametes, they can mix and match. So you could have a big R with a big Y in a gamete. You could have a big R with a little y. You could have a little r with a big Y. Or you could have a little r with a little y. These guys can mix and match to form gametes. Now, if you're doing a genetics problem on your exam and you're trying to figure out how to form these gametes, the way that you would do it is similar to the way you would do a math problem. I don't know if you've ever heard of the FOIL method, which stands for first, outer, inner, last. So you take the first R and the first Y. There you are. Then you take the outer, the first R and the outer Y. There you go. Then the inner, these two here, boom and boom. And then the last of these, so the little r here and the little y here, okay? So that's how you would get the gametes to put on this massive Punnett square here. And we're gonna do the same here, because we're crossing two of these together, remember, for our F2 generation. Now, I'm going to let you fill this in on your own. And the way you would do this is, for example, this square would be big R, big Y, big Y. And this one would be big R, big Y, little y. And this one would be big R, oops, little r, big Y, big Y, big R, little r, big Y, little y. Okay, if you do this all across and then you count up the phenotypes, if you did this all throughout and you counted up the phenotypes, here's what you'd get. Okay, if you're talking about round and yellow, this is just physical appearance, not genotypes, phenotypes. So remember that this is a dominant phenotype and this is a dominant phenotype. If you were to count up round and yellow outcomes here, you would find that 9 of them fell into that category, okay? If you counted up wrinkled, I'm going to abbreviate that, and yellow, okay? This is a recessive dominant. You would find 3 fall into that category. If you were to count up how many were round and green, okay, so that would be dominant recessive. 3 would fall into that category. And if you were to count up how many were wrinkled and how many were green, so that's recessive, you would get 1. This explains Gregor Mendel's 9:3:3:1 phenotypic ratio, phenotypic ratio that he observed when looking at two traits at once, okay? So what is he really observing? What he is observing in this phenomenon here, where these gametes can mix and match, he is observing something called the principle of, and we've talked about this in our previous chapter, the principle of independent assortment, okay? So in other words, remember that we talked about, here's your maternal set of chromosomes, here's your paternal set of chromosomes. Let's say that we have a big R here and a little r here and a big Y here and a little y here, okay? If you were to go through this whole process, right, you would end up with a situation, let's see here. So let me back up. So here we have our homologous pairs. They've lined up during meiosis, okay? And we have all the maternal on one side and all the paternal on the other. And for some reason, they've swapped them out here. So let me fix that. Little r, little y, big R, big Y. And let's just say you go through your meiotic division, okay? So you can have little r, little y, little r, little y, little r, little y. Here we'd have big R, big Y. You get where I'm going. But what if during meiosis, when the chromosomes migrate to the middle, so this is metaphase 1 of meiosis, where the chromosomes migrate to the middle, what if you have paternal, maternal, and then maternal, paternal? Do you see what's happened here? In other words, the way that the chromosomes line up in this phase is random. And that's going to give us a different combination in the gametes. So if this was little r, and this was little y, and this was big R, and this was big Y, satisfy yourself that at the end you would end up with this combination here. And you can go through and you can change the way that these line up, and it would correspond to these possible outcomes here. So this is independent assortment, okay? This is basically what Gregor Mendel discovered, but remember, he didn't know about meiosis. We can now explain Gregor Mendel's observations because we now understand meiosis. So let's summarize the principles or the big ideas that Gregor gave us about inheritance. And it turns out that they're all related to meiosis. Okay, so number one, there are 2 copies of each gene. Oops. All right, that's diploidy. Number 2, he discovered something called the principle of segregation. In other words, the 2 factors separate. And if you recall, the 2 factors separate during meiosis 1. Meiosis 1 is our reductional division, right? Specifically during anaphase 1 of meiosis 1. The next thing that he gave us was this principle of dominance and recessive, also principle of dominance. Oops. And then the fourth thing he taught us is the principle of independent assortment. Ooh, it does not like it when I try to write fast. Hang on. There we go. Let me fix that. My tablet is not being kind today. independent assortment. And that is what happens during metaphase 1 in meiosis 1, where the chromosomes line up randomly. Okay, so these are the basics that we learned from Gregor Mendel on how inheritance works. The next part of the chapter is going to talk about relating these ideas to chromosomes, which we already have, and then looking at some exceptions to, or maybe some extensions of Gregor's big ideas. So talk to you soon. Bye-bye.